Hello, and welcome to this webinar titled Understanding Prebiotics and Probiotic Mechanisms that Drive Health Effects, organized by ILSI Europe and the International Scientific Association for Probiotics and Prebiotics. My name is Mary Ellen Sanders. I'm the Executive Science Officer for ISAPP, and along with Michaela Miani, a Scientific Project Manager of ILSI Europe, we will moderate today's webinar featuring top experts in the fields of probiotics and prebiotics. But before we get started, I have just a couple of general announcements. We will be running a live question and answer discussion after the presentation. You can submit your questions and comments in the Ask a Question feature at the bottom of your screen. If you have a specific question for one of the speakers, please be sure to write that speaker's name before the question. All the webinars will be recorded and made available through the registration page on both ILSI Europe and ISAPP pages. So without further ado, I would like to introduce Karen Scott, Senior Research Fellow at the Rowett Insititute, Aberdeeen University in the UK, and Koen Venema, Professor of Gut Microbiology at the NUTRIM School of Nutrition and Translational Research and Metabolism in Venlo, the Netherlands. Today, Karen and Koen will be presenting about prebiotics, their mechanisms, and how they drive health benefits.
So the presentation on prebiotics, how they work and the impacts they have on health will be split into two parts, presented by myself and Koen Venema. I want to start by making you think about this question. Which of these could be classed as a prebiotic? A milk-based product containing live bacteria, a refined sugar consumed as part of the diet, or an indigestible dietary carbohydrate? Although most people know what a probiotic is, with an example being answer A, fewer people know that a prebiotic is not answer B, a refined sugar, but is actually answer C. Indeed, if we look at the definitions for probiotics and prebiotics from recent ISAPP consensus panel documents, bringing together groups of eminent scientists to come up with valid, accurate definitions, we can see that probiotics are live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. And in contrast, a prebiotic is a substrate that's selectively utilised by host microorganisms, conferring a health benefit. So basically, probiotics are added live bacteria, whereas prebiotics are food for resident bacteria. And these are some examples of foods containing probiotics or prebiotics. And many vegetables are actually sources of prebiotics, although the amounts they contain can be quite low. Now, the definition of a prebiotic, as shown here, does not limit prebiotics to only having an effect in the gut, but that's entirely what we will focus on today. And if we look specifically at the phrasing of the prebiotic definition, it's a substrate. So this means it's a substance as a food source for bacteria that are in the large intestine. It has to be selectively utilised by host microorganisms. This means that it can't be used by all bacteria, but should only be fermented by one or a few colonic bacteria that are beneficial. And it has to confer a health benefit. This is often the hardest part of a prebiotic definition because the health benefit has to be measurable and therefore confirmable. One example of a successful prebiotic is fructooligosaccharides, or FOS. These are growth substrates for bifidobacteria and some other colonic bacteria as well. And they have been shown to improve calcium absorption. So therefore, they have a measurable health benefit. Other accepted examples of prebiotics are shown here. And there are also several compounds that are classed as candidate prebiotics. And these compounds are often candidate prebiotics because they don't yet have sufficient defined evidence of their modes of action and the actual health benefit they can confer. when we think about the mechanisms of prebiotic action, bacterial fermentation is a very important aspect of this. As the bacteria use prebiotics for growth, they ferment them and release other products. And there are actually several options for product formation, some of which involve bacterial cross-feeding. If we think about the production of short-chain fatty acids by bacteria in the gut from prebiotics and other substrates, then you can get direct utilization of the prebiotic or complex carbohydrate by short-chain fatty acid producing bacteria producing butyrate or propionate, which are beneficial short-chain fatty acids. You can also get degradation of these complex compounds into shorter chain breakdown products, and those can then be used by short-chain fatty acid producing bacteria. Or you can get degradation of the prebiotic substrates by non-fermentative bacteria, say for example bifidobacteria, and this can produce intermediate products such as lactate or even the short-chain fatty acid acetate that are then used by gut bacteria. So these are examples of bacterial cross-feeding, whereby the intermediate products are actually substrates for other bacteria. And these are important mechanisms of action of prebiotics. The health benefits of prebiotics must be confirmed in good, reproducible studies in the target host. The effects can be local effects or systemic effects, and we're going to explain this further in this presentation. Whenever a food containing a prebiotic is marketed, the added dose of prebiotic in that food must be an appropriate dose. It has to be the dose that was tested in the trial. So I'm going to focus on the local gut effects of prebiotic substrates. Prebiotics, undigested in the upper intestinal tract, will enter the colon, and there are three immediate effects that they can have, or three local effects. Prebiotics are a type of fibre, so therefore they contribute to fecal bulking and increased transit rate through the colon. These are important factors protecting the colonic epithelial cells from any toxic compounds that might have been taken in the diet. And they also make the increased transit rate make sure that any such toxins move through the large intestine more quickly, again protecting the gut cells. Bacterial fermentation of the prebiotic causes a lowering of the pH. This helps to inhibit the growth of pathogenic bacteria, which don't generally like to grow in an acidic pH. And the lower pH also improves calcium solubility, and therefore uptake of calcium, and therefore bone health. Finally, two of the short-chain fatty acids that are produced by prebiotic fermentation, butyrate and also propionate, are important energy sources for the gut epithelial cells. This helps gut cell turnover and keeps the gut healthy. So these local effects have very specific health benefits. And I've shown this again here. And the first one of these, the improved gut transit rate and therefore stool bulking, actually has a European Food Safety Authority positive opinion and can be used as a health claim on foodstuffs. The other two health benefits shown here don't have such claims as of yet, but they have definitely got these proven effects. I just wanted to add that prebiotics are not just for use in humans. They're also used in companion animals, again to try to improve health, and also in farm animals, where they are used to promote the growth of the animal instead of antimicrobials. And this is a very important use of prebiotics as well. I will now hand over to Koen, who will discuss the systemic effects in more detail.
All right, so as Karen Scott just mentioned, I will discuss the systemic effects of prebiotic fermentation by bacteria. as indicated here. So this actually is a list of health endpoints in human trials of orally administered prebiotics, which have been split up in studies that have effects on the metabolic effects, studies that look at satiety and immune modulation. And then most of the studies that are listed under other have been discussed by Karen. So you can see that there are actually a number of different health endpoints that have been addressed by ingesting oral prebiotics, where the effects are actually accomplished through fermentation by the gut bacteria, and then through uptake of metabolites or through interaction with the host, there are systemic effects that are accomplished. Now, one of the first things that I'd like to address is effects on metabolic effects and satiety and the benefits attributed to prebiotics. To start off that discussion, I show this overview that has been published recently, where here at the bottom of the slide, you see that food ingredients, particularly dietary fibers and prebiotics, are fermented by the gut microbiota into these short-chain fatty acids. Part of these short-chain fatty acids are used as an energy source by the epithelial cells, which is this local effect that Karen discussed, but also part of the short-chain fatty acids actually end up in the bloodstream, where they go to the liver, which actually extracts most of these short-chain fatty acids. And there they are involved in gluconeogenesis, for instance. But also part of those short-chain fatty acids reach other organs like the brain, muscle, and adipose tissue. And in all of these different tissues, those short-chain fatty acids may elicit a particular effect. Now, they do that through interaction with receptors that are present in these organs. And here it is only drawn on the epithelial cells, but these same receptors are present in the other organs. These are G protein coupled receptors. There are two that interact with short-chain fatty acids, 41 and 43. And for instance, they lead to the secretion of these satiety hormones, PYY and GLP-1, which then travel to the brain and lead to satiety. On the other hand, in liver, adipose tissue, and muscle tissue, they may actually lead to other beneficial effects in these organs, which ultimately leads to a lowering of inflammation. We'd like to target fermentation of carbohydrates to the distal gut, and this slide indicates why. So if you look at the fermentative capacity of the gut microbiota in the proximal colon here, we see that primarily carbohydrates are being fermented there. This is partly because microbes would like to incorporate any protein that is present in the meal into their own biomass, into their own enzyme machinery. But as they travel along the gut, fermentable carbohydrates become depleted, and in the distal colon we see the onset of protein fermentation with concomitant production of toxic metabolites. Now, also in the distal colon, there usually is the onset of colon cancer and inflammatory bowel disease, which then subsequently may spread throughout the rest of the GI tract to the rest of the body. So there seems to be a correlation between the two. So wouldn't it be great if we could extend fermentation of carbohydrates towards the distal colon to prevent these occurrences of production of these toxic metabolites? So if we ingest a prebiotic that then makes it to the colon, it would be good if we could get fermentation in that distal colon. As I said, for increasing carbohydrate fermentation and then subsequently decreasing protein fermentation. But one of the other effects that we can understand is the fact that there is a higher density of these satiety hormone producing cells in the distal colon. And if we can get those cells to increase the secretion of these hormones, we will induce satiety, and that of course will have an effect on food intake and hopefully in the end also on body weight. Now that's with respect to obesity, metabolic effect, and satiety. What about the immune system? Well, this is a simplistic schematic of the immune system where people thought that it is a balance between inflammatory and a antibody-producing side of the immune system. And that balance can be disturbed, and preferably, of course, it is not. Now, I said a simplistic picture because the balance is actually much more intricate than is shown by the first one. I particularly like this one, where we hope to keep our immune system in check. But of course, with infections and things like that, and allergy, that balance is disturbed. Now, what happens if we use prebiotics and interact with the immune system? Well, the immune system contains what is called antigen-presenting cells, and this is a picture of a dendritic cell over here. And those cells have a lot of different receptors that can interact with dietary components or microbial components. Now, to focus on receptors that interact with carbohydrates, some of these are listed here. It has been shown that, for instance, the probiotics beta-glucan or fructooligosaccharides or galactooligosaccharides or pectin have been involved in interacting with these immune cells through these receptors. And that then subsequently led to the balancing act with the immune system. Now, this has been shown in infants, adults, and elderly. So in this figure, there is an overview of prebiotics interact with the host immune system. They can do that by activating or blocking host receptors that are involved in the immunological response by directly binding to these receptors. Or, as we saw, they can modulate the composition of the gut microbiota, the production of bacterial metabolites that can subsequently interact with the immune system. Also, there can be a direct interaction with bacteria, by which adhesion of these bacteria to the mucosa and uptake by bacteria by the host may actually be modulated. And if these bacteria normally interact with the immune system, of course, that interaction is also changed. So there's a number of different mode of actions that has been shown on this slide. In the intestinal epithelial cells, this receptor-mediated interaction that is indicated may be important. I mentioned these dendritic cells, this antigen-presenting cell. What is interesting about that is it can stick its dendrites through the epithelial gut lining, and by that actually sample here what is present in the gut, right? So there's a number of intricate mechanisms how the immune system interacts with what we eat and with our gut microbiota. So if we sum that up into health benefits attributed to prebiotics in systemic effects, then also in this particular case, we have one ISAPP positive opinion on the effect of prebiotics. And this is on lowering glycemic index, lowering your blood glucose levels, which of course is related to the metabolic syndrome and effect to that. So the health benefits are it increases insulin sensitivity, decreases lipid accumulation, it increases the barrier function of the gut, which is a local effect, but the effect of that is that there is a lowering of translocation of bacterial products, which leads to a lowering of inflammation systemically. Then with respect to satiety, studies have shown an increased production in these satiety hormones, PYY and GLP-1, which leads to increased satiety and lower food intake. And then lastly, with respect to the immunomodulatory effects, the health benefits are there that these prebiotics may boost the immune system, lower inflammation, not only locally in the gut in inflammatory bowel disease, but also systemically, and may also lead to a lowering of allergic reactions.
Our next speakers are Colin Hill, Professor of Microbial Food Safety in the School of Microbiology at University College Cork, Ireland, and Sarah Lebeer, full Professor of Microbiology and Biotechnology, University of Antwerp, Belgium. Colin and Sarah will be presenting about probiotic mechanisms and health effects, from molecules to different levels of action.
Okay, so the title that I've given this short presentation is that it's all about molecules. And the very simple point I want to make here is that life, health, disease, everything really that happens in biology is about molecules interacting with molecules. And therefore microbes, whether they're probiotics or pathogens or whatever microbes you want to consider, can only affect a host through the immune system, enteric nervous system, epithelial cells, microbiome, whatever, by producing molecules which then interact with resident molecules. If that doesn't happen, then there can be no effect. And it's important to notice that these molecules could have been produced prior to administration. So the incoming probiotic, the live bacterium that is a probiotic, may have already produced the molecules that are going to interact with the host, or of course they could produce them in situ in the gut. And here's just an example of a bacterial molecule, perhaps the most potent bacterial molecule that we know of in science, which is the botulinum toxin. And literally in femtogram amounts, the production of this toxin by a bacterium can cause paralysis and even death of a human by interacting with very specific receptors on the human neurons. And so we accept, in this instance, for example, that small numbers of molecules produced by a bacterium can have very dramatic effects on the health of the host. What we're beginning to accept more and more is that microbial molecules can also begin to have, or can have an effect on the health of the host. So if we look in this simple graphic, we've got that health is an outcome of an interaction between host factors and the microbiome. Now the host factors are almost certainly the most important: genetics, things that influence the host, but we do believe more and more, and particularly in certain chronic diseases, that the microbiome plays a very important role. And the diseases listed on the bottom of the slide are some of the ones for which there's the best evidence. Now we know that we can influence host health by, for example, pharmaceuticals, our lifestyle, our diet, and we can influence the health of the host. We also know that probiotics can influence the health of the host by directly interacting with the enteric nervous system or the immune system. But another avenue by which now we can see that we could perhaps interact or influence the health of the host is by influencing the microbiome. And on this side, it opens up a lot of possibilities. We've still got diet and pharmaceuticals and lifestyle and probiotics, but now we have things like prebiotics, which you've heard about from Karen and Koen, things like fecal microbiota transplants, bacteriophages, antibiotics, lots of different ways that we could influence host health via the microbiome. But once again, it all has to be done through molecules interacting with molecules. I like to make a comparison between pathogens and probiotics because I also work on pathogens in my lab. And again, we accept without question that pathogens can influence the health of a host. They're infectious pathogens, at least, are living bacteria, which are required then for the physiological effect, which is mediated by molecules. And we have a much better understanding of some of the molecules involved in pathogen infection. Probiotics also are living bacteria, which have a physiological effect mediated by molecules, but the evidence perhaps is not always as compelling. So using pathogens as an example, certain pathogens like enterovacive E. coli, for example, they produce surface structures like these type 3 pili, which then inject proteins into surface cells, causing rearrangements, the uptake of these pathogens, influencing the immune system and leading to disease in the form of diarrhea. But other forms of E. coli can secrete molecules into the lumen of the gut, which then interact with the epithelial cells and again lead to diarrhea. And probiotics can do these both of these things as well. They can interact directly with the immune system, the epithelial cells, or they can secrete molecules that have an influence on host health. Unfortunately, this is always complex. In the case of enteroinvasive E. coli, for example, there are at least 50 molecules involved in the invasion and the disruption of the host health and causing disease. And probiotics are likely to be similar. Probiotics are certainly produce multiple molecules. These can all interact with the microbiome, with the host, at multiple points. You're going to get lots of downstream effects. And so it's not very simple. And it requires a lot of research to find out what molecules are important. And as a result, we very often use animal models, tissue culture models, organoid models. And, you know, this is somewhat unsatisfying that we're not working very often with humans when we're trying to identify mechanisms. The best case scenario, the most simple scenario, kind of a Cox postulates for probiotics, would be where the entire probiotic effect is due to a single molecule. And then you could either eliminate that molecule, knock out the gene that produced that molecule, or just use the molecule itself. But that's a relatively rare event. And what I'm going to do is give you some examples, just a couple of slides, where individual molecules are largely responsible for probiotic effect, and we've been able to demonstrate that. And then Sarah will pick up on that in her section. So here's one example from our own lab where bacteriocins, small antimicrobial peptides, actually act as a probiotic factor. So here you see 6 mice representing 6 groups of mice. Each one has been fed a probiotic bacterium and then infected with Listeria monocytogenes. And Listeria infects the animals. That's the light you can see here. The Listeria were lux tagged, and they cause what is normally a lethal infection for these animals. You can see that one of the animals is completely resistant to this Listeria infection. And obviously all the animals in that group were completely resistant. And when we looked at that particular probiotic, which we now know to be what we used to call Lactobacillus salivarius and now is Lactobacillus salivarius, UCC118, we can see that it produces a bacteriocin which can kill Listeria. So this was an obvious potential mechanism. There's the bacterium producing a bacteriocin that kills Listeria, which is this lawn of bacteria. So we generated a knockout, which you see here, a colony which can no longer produce the bacteriocin. And when we look, we find that the colony that no longer can produce the bacteriocin can no longer protect the mice. So here's control mice infected. Mice given the probiotic are completely protected, but then mice given the probiotic with just a single gene knockout are now equally open to infection. So the probiotic effect here is due to a single molecule. So it is kind of a dream scenario for probiotic scientists. Here's another example, also from Cork, but from Dawa Van Sinderen and Mary O'Connell Motherway. And what they showed was the bifidobacterium. You see a bifidobacterium here. They produce pili. And you might be able to see the pili here. They've been immunogold labeled. And you can isolate the proteins that make up this pili, and you can isolate those and purify them. And what Mary and Dawa have shown is that this pili structure is required for bifidobacteria to colonize conventional mice, but then it also drives cell proliferation, which you can see here. It doesn't matter if you can't see the actual staining, but what this shows is that you get tissue proliferation or wound repair in the presence of these bifidobacteria. It does not happen in a knockout that can no longer produce the pili. And if you just take the pilin protein itself, the purified pilin protein, you can recapitulate this effect. And so we now see once again that an effect of a probiotic can be reduced down to the effect of a single molecule. Another really nice example from Patrice Cani's lab in Belgium is showing that a bacterium called Akkermansia mucinifera can have an effect on metabolic syndrome. We can do this in mice, and also it's been demonstrated to do this in humans. And so the live bacterium has this probiotic effect that you can see in both humans and in mouse models. But what they showed in this really nice paper in Nature Medicine a few years back was that actually pasteurized Akkermansia can have the same effect, or maybe even a slightly improved effect compared to the live bacterium. So obviously the effect must be due to molecules that had already been produced by the Akkermansia. And in fact, in a really beautiful study, they were able to define the protein AMOC 1100, which actually causes this effect. And again, these graphs confirm that, and I'd urge you to read this paper if you're interested in this study. But once again, a particular protein is the probiotic factor in this case. There's a very well-known example, of course, that people who cannot digest lactose can consume yogurt, and that was linked to the probiotic effect of the yogurt cultures, because the yogurt cultures contain an enzyme, lactase, which can break down the lactose, and that's the reason why people who can't digest lactose is because they don't produce sufficient amounts of the enzyme lactase. And once again, you can recapitulate the effect of the yogurt cultures by the enzyme on its own. So a really nice example, again, of reducing a probiotic impact down to a single molecule. And the benefits of doing this, of course, is that if you know which molecule or molecules are responsible for the probiotic effect, you can do much better strain selection strategies if you're trying to develop new probiotics or improve existing probiotics. It will also inform therapeutic approaches. If you know two different probiotics work by two distinct mechanisms, you could maybe combine those probiotics to treat a particular disease. While if you don't know the mechanisms, you won't know the benefits of strain combinations. It should obviously allow much more precise dosing. If you know the mechanism, you know how much of that molecule you will need to elicit the health effect. And hopefully, knowing mechanism isn't just an academic pursuit. It will genuinely lead to more efficacious drugs, food supplements, or food ingredients. And that's the hope for all probiotic science, that of course we will improve probiotic or improve human health. So now I'm going to pass over to Sarah Lebeer, who's going to continue on this topic.
Okay, so after the presentation of Colin, I will also talk about probiotic mechanisms of action, but I will want to start by looking at the mechanisms from a host perspective. And in this review paper shown here, which was actually an outcome of an ISAPP discussion group, we categorized the main mechanisms from a host perspective into 5 main categories. And as you will notice, actually these categories, they reflect also many of the health benefits mediated by prebiotics. So the first, actually often the most important mode of action is modulation of the composition and activity of the indigenous microbiota, and at least temporarily. So this can be, for example, by the production of lactic acid, which can then be consumed by lactate utilizers and result in butyrate, for example, as nicely presented by Karen. Lactic acid can also be directly antimicrobial and have an impact on certain gut pathogens such as salmonella. A second major mechanism is enhancement of the epithelial barrier function. For example, the Pili presented by Colin, the Pili of bifidobacteria, they are known to enhance the barrier function of the gut. The third main mechanism of action is modulation of the immune system. This is often mediated by different cell wall molecules of probiotics interacting with different immune receptors. And Koen has already given a very nice example of all the different host receptors that can interact with microbial molecules, but also prebiotics and metabolites. And the fourth main mechanism is modulation of systemic metabolic responses. This can also be mediated by probiotics, and for example, this includes effects on blood sugar levels or blood lipids or even cholesterol levels. And then the fifth mechanism is signaling via the central nervous system. This can be the effects on the gut-brain axis. For example, this can be because probiotics can produce certain neurotransmitters such as histamine or GABA, and in this way have an effect on anxiety, for example, or depression. For more than 15 years, I've been working on Lactobacillus rhamnosus GG, or Lacticacea-Bacillus rhamnosus GG, as we name it now, which is one of the probiotic strains with the most widely documented health benefits through various well-controlled clinical studies. And actually, yes, if you look at all the different health benefits that have been documented for LGG, it's quite apparent that you can also categorize them into these 5 main modes of action. So there are clinical studies showing a benefit on modulation of the microbiota. And in this way, it can benefit gastrointestinal symptoms. Also, other studies have shown that they can promote oral health or reduce the need for antibiotics. A second major effect of LGG is that it can enhance the epithelial barrier function. This is also related to the promotion of gastrointestinal health, but it can also reduce, for example, leaky gut, and that's also linked to reduction of eczema risk in certain families with increased susceptibility or decrease of food allergy. The third main mechanism of LGG is also a modulation of the immune system. This is, for example, also linked to this reduced eczema risk, but also LGG has also been used as vaccine adjuvants. A fourth main mechanism is again related to modulation of systemic metabolic responses. For example, LGG has been shown to reduce metabolic syndrome or in pregnant women, and also it has been shown to improve blood lipid levels. And then the fifth mechanism is related to signaling via the central nervous system and the reduce of anxiety and stress. But these mechanisms, they are not all mediated through the same molecules. So during the past 15 years, one of the major questions in my research is how is LGG able to exert such a wide variety of health benefits? Is it all through different molecules or are similar molecules, and how can we really explain these effects? And in this figure, this is actually also an outcome, but now from an ILSI probiotic expert panel. We have tried to make a translational pipeline that can help us answering this question. And as Colin has shown in his presentation, he has mainly focused on different probiotic effector molecules and how they can predict certain health benefits. For LGG, our major questions were centered to the fact that it's so clinically well documented and that we want to better explain how it's able to exert all these different health benefits. So we have been running a number of different trials. So in order to answer this question, we have built up a huge toolbox to identify the specific mechanism of action of LGG. During the past 15 years, we worked on genome editing tools and the construction of knockout mutants, which are important, as Colin already said. We have optimized various biochemical tools to extract all the different cell wall and other bioactive molecules. We have worked on phenotyping. We have developed several mini models. Recently, we're also including a lot more comparative genomics to better explain what are the strain-specific molecules of LGG compared to more conserved properties. We're increasingly using microbiome analysis to really look at how it can survive in different human body sites. And we also are running our own human intervention studies with a focus on unraveling modes of action. And based on all these different tools and analysis, we actually now know that LGG has a whole toolbox of different molecules and metabolites and enzymes that can help to exert its health benefits. And this includes its robust capacity and its good growth capacity, which enables this large compendium of metabolites and enzymes to be active inside the human body. It also has a very thick cell wall with lots of very active immune and epithelial signaling molecules. It produces lactic acid, which is a basic metabolite which has very important antimicrobial activities, but recently we also know that it has some immune modulating activities. It produces Pili, which are crucial for adhesion. We'll come back to that in the next slide. But it also produces enzymes such as the major secreted protein 1 and 2, which have a key role in epithelial barrier function. And it has this special lipoteic acid, which has an important immune signaling role. But perhaps the most special properties of LGG are its PAS-CBA Pili. And as you can see here on the second picture, these are the thread-like structure, especially at the poles of this rod-shaped bacteria. And these PCBA pili, their main function is promoting adherence to the mucus layer and epithelial cells, and they actually function as a kind of zippers. So they promote close contact with the epithelium, and then they can really bring the LGG cells close to the epithelium like a zipper. And we managed to construct different knockout mutants of the pili, and that you can really show to pinpoint certain role of the pili in different properties. So here you can see the wild type and how it adheres to the epithelial cells. And when we remove the pili, by this creation of a knockout mutant, you can see that the adhesion capacity is almost completely lost. And then in another mutant where we removed the EPS genes and we saw increased exposure of the pili, we saw increased adherence. We also used blocking antibodies, and then we also were able to block the adhesion. So by this, using these knockout mutants, we were really able to show that the pili had a key role in adhesion. And this was in cellular models, but we also wanted to validate the role of the pili in different, more preclinical and animal models, and also host physiological relevant models. And for this, we also give our mutants to different other labs around the world. Here's, for example, a very nice study of our colleagues in the United States who used our pili mutant to study the effect of LGG against protection against radiation-induced injury. And as you can see, the wild type was able to reduce apoptosis, and this activity was much reduced in the pili mutants. So the pili have some role in this preclinical model. However, when we, in my own lab, we were studying the effect of LGG in DSS colitis, we actually saw no major impact of the pili mutation. So it's clear that the pili have a modulating role, but they're not the crucial molecules, or definitely not the only molecules with anti-inflammatory effects. But of course, these are only animal models. So the next step is to validate the mechanism of action in humans and in clinical trials or in the other targeted hosts. And here you can see, here I want to mention the work of Willem de Vos, who has actually managed to create non-GMO pili mutants, or actually to be able to select non-GMO pili mutants, which you can use in human volunteers. And I know 2 of his studies have not yet been published, but 1 study has been published in Frontiers in Immunology. It was a study together with Glenn Gibson, also an ISAPP board member. And here they used LGG wild type and the pili mutant, which is a PB12 strain, together with a symbiotic, together with a prebiotic soluble corn fiber. And they tested them in elderly, and they tested different parameters. They saw some benefit of the wild type, especially for total cholesterol and LDL cholesterol, which was not present for the mutant, but it was only in a subgroup which has already elevated total cholesterol levels. They didn't see any difference for LGG wild type and mutant for immune parameters such as NK cell activity and microbial modulation. So again, also in humans, the pili seem to have some effects on the health benefits, but only a minor modulating effects. So with this example, I really wanted to highlight that we should be very careful with probiotic research on mechanism of action to have this reductionist view. Based on all the examples I saw so far, I think it's always there's always a complexity of modes of action. So there's always different molecules that act together to mediate health benefits. And in the case of LGG, its pili are important to promote adhesion and host contact, but also its high lactic acid production are important. Its major secreted proteins, its lipotechic acid, its CpG DNA, and its robustness and its survival capacity to withstand several different stresses. And as I also highlighted in the beginning of my talk, I want to stress that we should also not forget the complexity of the host Every individual has a different gut microbiota composition, a different host physiological state, a different health state, and it's very important that we stratify patients into responders and non-responders. And I think this last example with LDG wild type and spontaneous mutant in elderly with elevated blood lipid levels is a nice example of the importance of stratification. Another well-known example now is this study with LGG versus placebo in acute gastroenteritis in children, where despite previous studies and meta-analysis showing benefit for acute gastroenteritis, they were not able to see a benefit. But if you look at the children that were included, it was actually a very heterogeneous population with different forms of gastroenteritis and different forms of pathogens involved in this gastroenteritis. So it's very important that we collect as much biomarkers on health status as possible. Better explain the benefits and better explain and understand the benefits of probiotics in different conditions. So I would like to end my presentation by highlighting the complexity of probiotics, because just by definition they're complex. They're live microorganisms that, when applied in adequate amounts, confirm a health benefit to the host, and they produce a lot of different enzymes and metabolites, and they have different CpG-rich DNA, but also their cell surface has very different molecules. They all have peptidoglycan, but then they can have different polysaccharides, different proteins, and different adhesive molecules. If you want to know more about the different molecules that have already been identified with a probiotic mode of action, I will refer you to this ISAPP discussion group paper with more details on the different molecules. And I would like to end with a few take-home messages. The first one is, it merely refers to myself. I think when I started this work, we were a little bit naive that we thought we would be able to identify specific molecules of probiotics that could be effector molecules, just as pathogen valence factors, but we now understand that it's much more complex. Different molecules interact together to exert health benefits. And I think for the future, we all should embrace this complexity better and really manage the complexity. So we should collect as much data as possible. I think now with all the systems biology tools and all the different high throughput molecular methods, we will be able to better do that. Because I want to end by saying that the beauty of probiotics is actually that they act by different molecules and a multitude of mechanisms of action. Thank you very much, and I think it's now time for a Q&A session.
Many thanks, Colin and Sarah. And to the audience, please remember to ask any questions through the question chat function. So we're now going to be able to move into our Q&A session. We have already received a few through the chat function, but please keep sending them through and I can direct them to the speakers. Please remember to add their name so that we know who the speaker is, which speaker you're targeting. So we have some great questions to get started. Karen, I'm wondering if we can start with you about a question about cross-feeding. Someone did not really understand the cross-feeding concept. Can you explain a bit more about the importance of bacterial cross-feeding?
Okay, so I guess cross-feeding is when one bacterium feeds off something that's produced by a different bacterium in the gut. And it can be cross-feeding on breakdown products of a carbohydrate. So you have some bacteria that are very good at degrading long chain carbohydrates and other bacteria that can only use very short chain oligosaccharides or disaccharides. So the bacterium that degrades the long chain releases all these little products that other bacteria can use. And that's one example of bacterial cross-feeding. So it's basically when one bacterium uses products released by another bacterium.
Great, thank you. There's another question that came in, and this one's for Sarah. Sarah, can you comment on how LGG works, or what is the mechanism of action that allows it to reduce antibiotic-associated diarrhea?
That's a very good question, but a very difficult one to answer. It's one of the best documented health benefits of LGG, and it's one of these examples that we don't really know why this is the case. I always think it's based on its three main activities. So there's definitely an antimicrobial activity playing a role. Maybe it's just its lactic acid production that can be directly antipathogenic for certain Enterobacteriaceae, or it can, like in Karen's example, maybe the lactic acid also stimulates butyrate producing bacteria. But then for LGG, we also know that it has a very good capacity to promote epithelial barrier function, and I think that's also important to prevent antibiotic-associated diarrhea. And it also has an immune modulatory function that might also be involved. But to make my answer shorter, I think the correct answer is we don't really know. We see the physiological effects, but it's probably not through one single molecule. It's a combination of different effects.
That's a great point. Okay, thank you, Sarah. And then for Koen, can I ask a question? Is there an established dose threshold for prebiotic effects? So and specifically, do you think that small doses can be effective, or are larger ones needed?
Yes, that's also a very interesting question. Usually doses of around 5 to 10 grams are being used in clinical trials to establish the effective or the effect of the prebiotic. There's been some studies that looked at lower doses, but very few studies that actually have done a dose response. And I think those are really needed to be able to see, how low can you go, if you like. There's also a certain maximum. If you ingest too many prebiotics, then you might actually experience issues like bloating and things like that. So I think, like Sarah, this was a long answer. The short answer is that we should actually do more studies on this to establish if there is a certain lower limit of a dose that is effective, and that could still lead to that prebiotic activity.
Okay, thank you. And this question's for Colin. You specified the complexity of probiotics and how they act in the human system. So the question has to do with being sure that animal models can really predict or replicate the beneficial effects that we are hoping to see demonstrated in humans. And are we oversimplifying this, or is it possible that single molecules that might be responsible for effects in an animal model really won't show up in a human model or a human system?
Yeah, I think that again is a very important and interesting question. I think with health effects, obviously we have to demonstrate the health effects in the target host. So if we want to make a claim that probiotics have a health effect, we can't demonstrate it in an animal and then assume it will transfer to a human. We have to demonstrate it in the human. But if we want to go after mechanism, we almost cannot do that in humans, because we can't use humans as experimental models, and we can't use genetically engineered knockout strains and overexpress genes and so on. And so we're stuck with animals for mechanistic insights and humans for the health benefits that accrue from those mechanistic insights. Now, of course, if you're not replicating the exact health benefit in the animal, then how do you know if you're really looking at the right mechanistic mechanism that's underpinning that? But I think in general, if you can show that an animal model of disease that replicates some of the symptoms of the human disease, and you can identify mechanisms that affect and ameliorate those symptoms, I think there's a good chance they will be actually the mechanisms responsible in humans as well. But it's a great question, and one I think that we'll always be stuck with. And we're stuck with in all aspects of medicine, really. I mean, all small molecules, they're all developed using animals to try and discriminate mechanism, and then humans for efficacy. So efficacy in humans, mechanisms in animals, I think.
That's a great way to phrase it. Thank you. And Karen, would you be able to comment on the symbiotic concept and what criteria are important in applying symbiotics?
Okay, thank you. Again, that's a good question. And in fact, there's a very recent paper come out this year from an ISAPP consensus book on this very thing about defining a symbiotic. But basically, our understanding now, or our interpretation now, is there are two types of symbiotic. One is a mixture of a probiotic and a prebiotic, and they, when delivered together, confer the health benefit on the host. But you can also get a type where the carbohydrate component of the symbiotic helps the probiotic bacterium to survive or exert the health effect. And the carbohydrate component within the symbiotic doesn't necessarily exert an effect by itself, but because it's needed to help the probiotic have the health effect, then that also is classed as a symbiotic. So there is a complementary and a synergistic types of symbiotic, and they're different, but they're both very effective.
Okay, great. Thank you. This next question is for Sarah, and it brings up a bit of a question from a regulatory point of view, but the question has to do with whether or not any of the health claims for LGG can be used on a product. Is the data strong enough to be able to do that?
I think, yeah, first of all, whether a health claim is allowed, it depends on the geographical region. So it depends on whether in Europe or in the United States or any other country. Then related to the scientific evidence and what we as scientists or as experts consider as enough evidence, it's very difficult to really pinpoint. Of course, it has to be documented in a well-controlled and good quality trial, and the statistics need to be good. For LGG, many studies have been done, but not all studies are that large. So it depends. And I think it also depends on the exact product formulation. Some studies have been done with LGG, but in combination with other strains and other prebiotics. So it really depends on the available evidence, whether you can state a health claim or not.
Okay, thank you. So this is a question for Koen. And it is an interesting question about the possible ways of collecting data for clinical trials. Do you think that there's value to including individual monitoring of health parameters, for example, using a smartwatch? And will that become a means for a scientific or a scientifically valid way to collect data in a trial?
Oh, wow. All interesting questions that are being asked. This is fascinating. I definitely think that these smartwatches, which are currently already being used in clinical trials, are an additional way of gathering data that can be used to interpret mechanisms or outcomes of pro and prebiotics, for sure.
Okay. So, Colin, here's a question that's going to require quite a bit of speculation. What do you think the expected next health benefits for apparently healthy consumers are on the horizon that will be able to be actually proven for a prebiotic or a probiotic effect according to EFSA criteria?
Yeah, that is a good question. If I had to guess, and it would be simply a guess, I would think probably in the area of antibiotic-associated diarrhea, where there's some really good evidence for probiotics having a positive benefit in reducing the duration, at least, of diarrhea, or maybe even preventing diarrhea in the first instance. And our IBS. I think the evidence for IBS as well, for certain types of IBS, is pretty compelling. If I was given my choice, though, I would love it to be one of the next generation probiotics. I would love to really have us push into that next generation and have a Faecalibacterium or an Akkermansia approved by EFSA, because I think that would really take the field into where it needs to go, which is not limiting ourselves just to the traditional probiotics.
Great. Okay, Sarah, I'm wondering if you can comment on this. If Lactobacillus rhamnosus GG is a model probiotic, do you think that we can extrapolate the mechanisms of action that have been identified for this strain to other strains or even other species? Or is it really important for the field to move forward to really identify new or additional model probiotic strains to start delving into?
Yeah, the short answer is that I think every probiotic has its own strain-specific properties, and it's always important to look at the specific strains and its property. So I used to always say, yeah, LGG is quite specific, and it has a specific pili, and it has its own unique property. While based on our newest research and also doing much more comparative genomic studies, I think there are really some core benefits or core mechanism of action of especially then lactic acid or lactobacilli, or now with the new genus names, it's a little bit more complex, but we see some core preserved properties that are important, like lactic acid production specific, peptidoglycan structure, lipotechic acid, or also, for example, these major secreted proteins. They're really conserved in the Lacticasia bacillus new genus. So it's not only LGG that has these specific properties. But on the other hand, and the second part of your question was whether I think new model strains of probiotics should also be explored. I think, yeah, that's definitely the case. But there are, of course, I think a lot of what Colin also has shown this bifidobacteria. There are also some very nice mechanistic and model studies on bifidobacterium probiotics.
Thank you. The next question is for Karen. You mentioned that bifidobacteria are non-fermentative bacteria. Can you explain that a little bit further?
Okay, sorry. So yeah, when I said that, what I was meaning, I guess, to say was that bifidobacteria do not ferment carbohydrates to produce propionate or butyrate. And this was previously talked about as being the reason why prebiotics worked, and that they stimulated bifidobacteria and raised the production of butyrate. Now, since bifidobacteria don't produce butyrate, it's not simple like that, and this is where bacterial cross-feeding comes in. However, bifidobacteria do metabolize carbohydrates, and they produce lactate, which in itself can be quite important in maintaining the pH in the colon. And also they produce acetate, and the acetate is often cross-fed on by other bacteria as well. But many bacteria in the human large intestine also produce acetate as a metabolic product. So they do produce acetate, and I probably should have said that in my talk. Sorry.
Okay, great. That clarifies it. And Koen, can you help us with a question? And it has to do with whether or not it's possible to prepare a prebiotic to be digested or released in different parts of the intestine. And if so, what are the best ways to do that? And do you know of any companies that are actually doing that technology?
It's an interesting but difficult question to answer. There's a number of technologies that could be used to get specific release of a prebiotic in certain areas of the intestine. Mostly, of course, prebiotics would target the large intestine, which contains this dense population of microbes. But even the large intestine can be split up in separate regions of let's call it microbial activity, where most of the fermentation of carbohydrates would take place in the proximal large intestine. But then as these microbes travel along the gut and fermentable carbohydrates become depleted, then in the distal colon they switch to protein fermentation. Now, protein fermentation leads to all kinds of toxic metabolites, so you would preferably want to prevent that. So to get a mechanism or tool to actually deliver prebiotics distally in the large intestine would in fact be beneficial for health, is what we think. So there are different ways of doing that. Mostly that is being explored with capsules that open up at specific sites in the intestine, either based on pH or based on microbial activity, in fact. So that is an area of research that is being carried out, but I don't think there are any products yet that are commercially on the market to allow specific targeting of prebiotics.
Okay, thank you. Colin, this is a question that has to do a bit with microbial ecology, and we've had several questions come in along this same line, and it has to do with, it has to do with how pathogens, how probiotics actually are able to exhibit antipathogenic activity, but also the flip side of that, which is, can any pathogens in the gut actually produce bacteriocins or other molecules that will counteract or negate the effect of a probiotic in its goal of harming or reducing pathogenic activity?
Yeah, I think a lot of our microbiology in terms of the bacteria that interact with the human body has been colored by our study of pathogens, infectious disease-causing pathogens, for, of course, decades before anyone really seriously studied probiotics. And so any factor which promotes the ability of a pathogen to cause disease was termed a virulence factor. And so we began to think of things that are, everything that helped a bacteria to cause disease is a virulence factor. But of course, some of those factors are merely helping the pathogen to survive and to grow and to colonize within the gut. They don't actually cause any damage to the host. And it's not surprising that probiotics share a lot of these characteristics because they share the same challenges. It's a challenging environment to live in the gut and to survive and to produce something and interact with the human body. That's a big challenge, and probiotics and pathogens share those challenges. I suggested a few years ago that we should discriminate between pathogen virulence factors, probiotic health benefit factors, and then what we should call niche factors, just things which help the bacterium to survive in that particular niche. And so it's not surprising that both pathogens and probiotics produce bacteriocins, they do, and that they compete with one another in that environment. And so yes, pathogens, probiotics, they share many characteristics, including producing bacteriocins, adhering to host cells, interacting with the immune system, but we should expect that.
Okay, and do you think that there's evidence that they would potentially inhibit a probiotic in their functions in the gut?
Well, probiotics aren't normal residents of the gut, so I presume the question means, can pathogens kill commensals in the gut? I don't think they'd be killing probiotics specifically. And yes, I mean, pathogens can produce bacteriocins which help them to establish a niche within the gut by killing commensals. That's definitely the case.
Okay. And here's a question actually that was asked to all the speakers, and so I'll let you guys decide who wants to take it first. But the importance of clinical trials for demonstrating health benefits is obvious in the field. How important is it, do you think it is, to determine the mechanisms of action that are behind a health benefit in order to have a health claim approved?
If I could start on that one, I think not at all. I mean, if you've got a probiotic that has an established health benefit that you can demonstrate in maybe 2 or 3 RCTs and, it's gold standard evidence, then I don't think you need any mechanism. We don't have mechanism for quite a number of small molecules that are approved for different diseases and different chronic conditions. But I think if you really want to advance the field and pick better probiotics, pick better combinations, then we need to know the mechanism, because that's the way in which we will determine the likely better or next generation probiotic. But to generate a health claim, absolutely unnecessary, I would think. Maybe the others would disagree with me.
Does anyone else want to weigh in on that?
I entirely agree with what Colin just said.
Now, there's one caveat to that, of course, which is if we're talking about prebiotics, you actually do need some evidence of prebiotic mechanism that it's mediated by the microbiota. So, Karen, I don't know if you want to weigh in on that component of a prebiotic claim.
Well, I suppose for prebiotics, yeah, it's important that the prebiotic stimulates the metabolism in some way, either the growth or the production of metabolites of specific bacteria, and not all bacteria in the gut. That's the current definition of a prebiotic. So you would have to have some evidence that the prebiotic compound didn't enhance the growth of everything in there, because clearly you don't want it to enhance the growth of bacteria that are potential pathogens, for example.
Okay. All right, great. There's a question here for either Colin or Sarah. Maybe both of you would like to weigh in on it. And it has to do with quorum sensing. And I think it's basically asking for clarification on what it is and what can actually initiate when or what causes this to initiate when two microorganisms are communicating with each other. Maybe Sarah, could you start?
Okay, yeah, so quorum sensing is actually a mechanism used by many different bacteria that they are able to measure a certain quorum. So they measure certain thresholds of their own population, and if they reach a certain threshold, they start to express certain genes that enable them to colonize or adapt to the new environment. So for many gram-positive bacteria, these are more peptides. For many gram-negatives, these are other types of molecules. And then you also have some more universal communication signals that can also be produced and that can be detected both by the strains or the species that produces them, but also other species. But most of the time, it are signaling molecules produced by specific species and only detect, or even specific strains, and only detected by the same clonal population. For probiotics, I actually did my master's thesis 20 years ago on quorum sensing, and I worked on the AI2 communication signal in lactic acid bacteria or lactobacilli. It's a universal quorum sensing mechanism. So the lactobacilli, they can produce AI2, and it can be detected also by pathogens. And we thought this signal could really mesh up with how pathogens use quorum sensing to express certain virulence factors only when they reach a sufficient density so that they can really overcome the immune system. But it was not so simple, that picture. So there's no real evidence yet that really it plays a key role in the gut, I think. Maybe, Colin, you can correct me, but I think it's a beautiful mechanism from a molecular point of view, but I'm not sure whether it really plays a big role in the gut and also in probiotic mechanism of action.
Yeah, I agree with you. There's very little evidence for that, but if I had to guess, again, I would say quorum sensing is probably very important, because remember the gut as well. We tend to think of the gut almost as we think of a fermenter or a tube, but it's actually a whole series of microenvironments. There'd be bacteria growing within mucus, bacteria growing in the lumen, bacteria growing in different niches throughout the gut. And I think what quorum sensing does is it gives bacteria intelligence. You know, an individual bacterium, I shouldn't humanize them too much, but they need to know where they are and who's around them. And quorum sensing gives them that level of intelligence. So some pathogens will only mount an infection once there's sufficient there to know that they will have an impact. And again, bacteria don't know anything and they don't think anything. But as a community, I think, in contact with other microbes and sensing biochemicals, including quorum sensing molecules, this is what gives microbes intelligence. And so I think it is really, really important. And I love the idea, and I wish it had worked out better when Sarah started talking about it, that we could drown out that conversation for pathogens, if you like, with probiotics. But I suspect probiotics don't reach sufficient levels, particularly in the colon, to drown out the signals from other bacteria. It's a great area for research.
Yeah. And I know we're getting close to running out of time here, but there's two quick questions for Koen and that I'm hoping we can fit in. One of them is, does food processing, such as cooking, change the availability of prebiotics? And then the second one is, do prebiotics have an effect on the human small intestine, or is it just the colon?
Again, great questions. I'm not sure if I'll be able to answer them within time, but to start with the second one, yes, prebiotics actually can have an effect also on small intestinal communities. We don't only have bacteria in the large intestine, there's also bugs in our small intestine. And yeah, there's no reason why those bacteria cannot ferment those. On top of that, for some prebiotics, we know that they actually interact directly with the host without the need to be fermented by the gut microbiota. So they may also have immunomodulatory activity. And most of that would actually happen in the small intestine, where there's actually a chance for these molecules to interact with the immune system, because as they hit the colon, I would say, with the high density of microbes that are there, they are relatively quickly fermented. Now, please remind me what the first question was. I forgot.
I'm sorry, was whether or not prebiotics were impacted by cooking or other processing.
Oh, yes. So, yes, definitely. Also, certain novel carbohydrate molecules that are being developed into prebiotics are, let's take pectin as an example, which really is affected by the cooking process. It is partly degraded, and then you end up with shorter molecules which will have a different effect on the gut microbiota than the original molecules that are present in raw vegetables. So the release from the food matrix is an important aspect of prebiotics as well. Now, most of the commercial prebiotics, they are purified molecules that are added to, like a dairy beverage or something like that. So they won't really be trapped in the original food matrix. But for certain novel molecules that are being developed towards prebiotics, this may certainly be the case.
Great, thank you so much. Well, unfortunately, that's all the time we have today for our webinar. I want once again to thank our speakers for their insightful presentations, and thank you to all the listeners for submitting some really great questions. It was a very good discussion, and I appreciate everybody joining us for this webinar. And as a reminder, a recording of the webinar will be available on the ILSI Europe website, which is www.ilsi.eu, and on the ISAPP website, which is isappscience.org. So goodbye, and thank you so much for your time today.